First, transesterification reaction. The 2' hydroxyl group at the branch point adenosine attacks the phosphate at the 5' splice site. This cleavage of the pre-mRNA at the 5' splice site results in the formation of a lariat structure, where the 5' end of the intron is covalently linked to the branch point A, and the 5' exon is released with a 3' OH group. This forms the C complex. Second transesterification reaction. The 3' OH group of the released 5' exon attacks the phosphate at the 3' splice site. This step cleaves the pre-mRNA at the 3' splice site, joining the 5' and 3' exons together and releasing the intron in the form of a lariat. This forms the post-spliceosomal complex. Spliceosome disassembly and intron degradation. The spliceosome disassembles from the mature mRNA, and the lariat-shaped intron is linearized and degraded by cellular enzymes. The mature mRNA is then ready for export from the nucleus and translation into protein. It's important to note that alternative splicing can occur where different combinations of exons are joined together. This process allows a single gene to produce multiple different mRNA isoforms and consequently multiple different proteins, greatly increasing the diversity of the proteome.
Spliceosome assembly. Several small nuclear ribonucleoproteins (snRNPs), which are complexes of small nuclear RNAs, snRNAs, and proteins, along with other protein factors, assemble on the pre-mRNA to form the spliceosome. This is a dynamic and ordered process involving several stages. U1 snRNP binds to the 5' splice site. Splicing factor 1 SF1 binds to the branch point. U2AF, U2 auxiliary factor, binds to the polypyrimidine tract and the 3' splice site. U2 snRNP displaces SF1 and binds to the branch point, causing the adenine to bulge out. This forms the A complex. The U4/U6, U5 tri-snRNP complex joins, forming the B complex. Rearrangements occur within the spliceosome, including the release of U1 snRNP and conformational changes in U6 snRNA, leading to the catalytically active B** complex.
mRNA splicing is a crucial step in the processing of pre-messenger RNA or pre-mRNA in eukaryotic cells. It involves the removal of non-coding sequences called introns and the joining of coding sequences called exons to form a mature mRNA molecule that can be translated into a protein. Here are the general steps of mRNA splicing, primarily carried out by a large RNA protein complex called the spliceosome. Recognition of splice sites. The spliceosome recognizes specific sequences at the boundaries between exons and introns. These include the 5' splice site, also known as the donor site, which typically has a GU sequence at the beginning of the intron. The 3' splice site, or acceptor site, which typically has an AG sequence at the end of the intron. The branch point, an adenine nucleotide located within the intron, usually near the 3' splice site. A polypyrimidine tract, a region rich in uracil and cytosine, located between the branch point and the 3'